This video we will discuss about this fluorochrome optimization for NFAT reporters for the activated T cells. So far we have optimized this number of NFAT binding sites and the promoter that is required for this reporter protein sequence, and we have come up to a point that this 8x NFTA binding sites gives us the most optimum signals intensity, and we can differentiate between non-activated T cells and the activated T cells. And this data box also gives us the signals, also give us the signals for the reporters proteins that will help us to distinguish between activated and non-activated T cells. So here we wanted to check which here the researchers has checked different fluorochrome or reporter proteins that will give the maximum signals for the activated T cells. And parallelly they also check these IL-2 productions to ensure that the T cells are actually activated. So when they did it they check it for the ZS green one, TD tomato, E2 crimson, and tag BFP2. So first they mention these IL-2 productions. We can see that IL-2 productions are there in all the 4 cases when they are stimulated with this anti-CD3 antibodies compared to the unstimulated case where the IL-2 production is almost negligible. Then they did it is they check the intensity of the fold change of different reporter proteins, and the fold change is compared between this unstimulated and anti-CD3 antibodies. And we can see that in the ZS green the fold change is quite high, dramatically high compared to the all other cases, where we can see that though we can see the difference between in the tag VF2, the difference in the activated and non-activated T cells, but the ZS green gives us the better signals. So they have decided to continue with this ZS green reporter proteins after the startup box sequence. And they have also shown this flow cytometry results, where we can see that when there is no NFAT construct, the reporter signals is almost 0. But in the case where NFAT construct are there and the cells, these hybridoma T cells are not stimulated, that is they are not treated with insulins or anti-CD3 antibodies, then we can see the signals is almost close to 0. But when this anti-CD3 antibodies are used for stimulations or activations of the T cells, we can see the increase in the signals to 87.9% in the case of ZS green proteins, and 18.1% in the case of TD tomato, 21.1% in the case of E2 crimson, and 70.8% in the case of tag BFP2. So in this case also it is confirmed that though the signals for this tag BF2 is also high, but the better signals they have got when they use this ZS green reporter protein. So now till now they have optimized 3 different things about their NFAT reporters for activated T cells. So first, what they did is they optimized the promoters that can be used, optimum promoters that can be used just before the sequence of the reporter protein sequence, that is data box, and they compare it with this mini CMV. And next they checked the signals at different numbers of NFAT binding sites. That is, they checked it for 4 NFAT binding sites, 6 NFAT binding sites, and 8 NFAT binding sites, and they find out that 8 NFAT binding site will give us the optimum signals. Now, and in this the results that are shown in this figure has compared the different reporter proteins that will give us the optimum different reporter proteins, and checked which can give us the maximum signals and can help us to distinguish between activated T cells and non-activated T cells. And they are able to find out that the ZS green one has the highest intensity fold change between the activated and non-activated conditions. So they decided to continue with this ZS green. So this is what they have so far optimized in their NFAT reporters uh, that can be used to check whether the T cells are activated or not. I hope you understood uh, the results that are presented here and how they optimize their fluorochromes for these NFAT reporters that is used for check whether T cells are activated or not. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.